welcome to class. Today we'll be looking at the October-November 2025 Igcse paper 2-1. So without much ado, let's get started. Question number 1. The diagrams show a plant at the start of an experiment and the same plant 2 weeks later. So you see the start of the experiment and you see 2 weeks after. Which characteristics of living organisms are demonstrated by the experiment? So the A option is excretion and growth, excretion and reproduction, growth and sensitivity, sensitivity and reproduction. Looking at the plant closely, you would see that the plant has grown. It has increased in size, so growth will be ticked, and then it's also tilting towards light. So we are safe to say that the 2 options you have there are growth and sensitivity. So option C becomes the correct answer. Question number 2. What can be used to classify organisms? So the first one is the number of bases in DNA. Number 2, or option B, the number of different types of bases in DNA. Option C, the sequence of bases in DNA. And option D, the sequence of bases in one protein. So you know that in biology, the modern system of classifying organisms, we now use the base sequences of DNA in classifying organisms and finding out similarities between them. Moving on to the next question. The diagram shows a single cell from an organism called Spirogyra. Which feature does Spirogyra share with plant cells? So if you look at it very well, a plant cell has a cellular cell wall and some other organelles. So let's pick them one after the other. A Spirogyra has a cell wall, and a plant cell also has a cell wall. So this is correct. A Spirogyra from the diagram has a chloroplast, and a plant cell also has a chloroplast. So this is also correct. A Spirogyra has a cytoplasm. You can see the cytoplasm here, and then here also a plant cell has a cytoplasm. That's correct. A Spirogyra also has this nucleus here. A plant cell also has a nucleus. This is also correct. And then finally, a vacuole is a major part of Spirogyra, and then it's also found in plant cell. So that is why option A is the correct answer to that question. Remember, the 3 parts you have in a plant cell that you don't have in an animal cell are your sap vacuole, your cell wall, and your chloroplast used in photosynthesis. Moving on to question number 4. The diagram shows an image of a chloroplast. Now the image is 5 cm long. The actual length of the chloroplast is 5 micrometer. This is micrometer, and then this is they give you the length, the image length in centimeter. So they now ask the question, what is the magnification of the image? So what you need to do here is very easy. It's not difficult at all, but the first thing you need to know is that you have to take them to the same units. You have to make sure that the units are the same before you find your magnification. So let's try to see how you can take that to the same unit. You know already that from our basic biology, 1 centimeter, 1 cm is equivalent to 10,000 micrometer. So it means that if they are giving you 5 cm here, 5 cm will also be equal to 50,000 micrometer. So doing that, putting that in the formula for the calculation, you would see that your magnification is simply calculated as what? Image size all over what? Actual size. Alright, so putting that in the formula, you would have magnification is equal to image size over actual size, and the image size is now 50,000. So you put 50,000 divided by the actual size or actual length, which was what? 5. So that will give you 5. And doing that very well, you have times 10,000 as the correct answer, which is option C. Okay. Moving on to the next question. A potato was used as a source of plant tissue. Okay. 4 potato sticks of identical size were cut from the same potato. Take note of that fact. Now, at the start of the investigation, each potato stick was 10 square long. The potato sticks were put into 4 separate petri dishes. Okay. Now, each petri dish contains a different concentration of sugar solution. The diagram shows the 4 petri dishes after 24 hours. Which dish contains a sugar solution that has a higher water potential than the potato tissue? Now, this is an application question, and this question is used to test your knowledge of osmosis. So osmosis is a condition where water goes from where it is in high concentration to where it is in low concentration, passing through what is called a partially permeable membrane. Now, when water enters into a cell, it is very clear that the cell is going to swell, is going to increase in size. So what they're asking you here is that which dish contains a sugar solution that has a higher water potential than the potato tissue? They said that each of the potato chip was 10 square long. So you have to look out for the diagram, find the one that has more than 10 squares at the end of the day, and that would be the answer. So let's count. This one has 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. So this cannot be our answer. This is out of it. So let's count this one and see. 1, because we are 10 square long at the start. 1, 2, 3, 4, 5, 6, 7, and 8. This is also not our answer. That cannot be our answer. 2, and then here you have 1, 2, 3, 3, 4, 5, 6, 7, 8, 9, 9 and a half here, almost 10, but it's not 10. So this cannot be our answer. Why? They told us that the condition on our basis here is that the dish contains a sugar solution that has a higher water potential than the potato. So water is going to flow into the potato cylinder. So let's count and see the last one. Here you're having 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Of course, this is the 10th, and there is this one that overlapped to become what? The 11th one. So that is why option D would be our correct answer. You should go and check up things like hypertonic. Check out solutions like this: hypertonic solution, hypotonic solution, and then isotonic solution. You can drop your understanding of them in the comments as we look through together. Moving on to question number 6. Which row shows correct statements about active transport? Take note, active transport. They give you 2 conditions. The first one is that they are showing the direction of movement, and secondly, they are asking if it requires a partially permeable membrane. Now, you should know that in active transport, substances move from where they are. Let me do this little illustration. So I have these little dots around here. I have several of them here, and I have just 2 or 3 of them here. When substances move from when they are in low concentration, they are low here, to when they are in high concentration, they need energy from respiration. These movements from region of the low to the region of the high is called active transport. So substances are moving against concentration gradients. If it were to be moving from where they are in high concentration to where they are in low concentration, in other words, moving from the high region to the low region, that would be diffusion. So the red represents diffusion, but the blue represents active transport. So let's pick it up in our answers to see. Moving down a concentration gradient, that is not correct. Let's find the one that says against a concentration gradient. So C and D are correct, but active transport requires a partially permeable membrane. So that's why the correct answer would be option D. Yes, it moves against the concentration gradient and requires a partially permeable membrane. That's also correct. So that's why D is the correct answer. Alright, so moving on to the next question, question number 7. The graph shows the effect of temperature on an enzyme controlled reaction. So you're seeing the rate of reaction on the y-axis and you're seeing temperature on the x-axis. Which statement describes the effect of temperature on this reaction? So let's see the statement that describes the effect of what? Of temperature on this reaction. Number 1, or the first option says, as the temperature increases to 25 degrees Celsius, the temperature speeds up. Is that correct? We're going to check. Option B says between 10 degrees Celsius and 20 degrees Celsius, the enzyme denatures. Okay, we're going to see that as well. Option C, between 35 degrees Celsius and 45 degrees Celsius, frequency of effective collisions increase. We'll check that as well. And the last one says the optimum temperature for this reaction is 40 degrees Celsius. So let's start with the last option. Optimum temperature for this reaction is 40 degrees Celsius. Optimum is where you have the highest point. From the graph, you can see that it is not at 40. The optimum temperature is around this region. So you see that it's at 30 degrees Celsius. So that cancels out this as our answer. Our answer, the D option, is wrong. Between 35 degrees Celsius and 45 degrees Celsius, the frequency of effective collisions increase. So let's see, where are you having the highest effective collision? 25, 35 and 45. 35 is somewhere around here and 45 is somewhere around, no, the reaction is already coming down, so that is also not correct. The third option is not correct. Between 10 degrees Celsius and 20 degrees Celsius, the enzyme denatures. 10 and 20, the enzyme denatures. Is that correct from our graph? That's not correct, because this is 10 and this is 20. There you are even having a very effective collision, so that is also not correct. And then the first one, as the temperature increases to 25 degrees Celsius, the reaction speeds up. That is the correct answer, because you're seeing here that you're having an exponential phase in this medium. At 10, between 0 to 25 degrees Celsius, that's where you're having the highest number of effective collision form. So that's why option A becomes our very correct answer. Moving on to the next question, question number 8. The graph shows the rate of photosynthesis of a plant in full sunlight in different concentrations of carbon dioxide. All other factors were kept constant. Now, what limits the rate of photosynthesis at 0.02 carbon dioxide concentration? Now, you must have learned in your basic biology that limiting factors are any factor that if it is not there, the reaction will not take place optimally. And for photosynthesis, there are 3 limiting factors. The first one is carbon dioxide concentration, the next is temperature, and the last is light intensity. So looking at the question they've asked us now, what limits the rate of photosynthesis at 0.02 degrees percent carbon dioxide concentration? This is where you have 0.02. Now, from the knowledge of limiting factors, as long as this graph, you see now that as you increase the concentration of carbon dioxide, as it's increasing, the rate is also increasing. So from this point of 0 to this point here, this whole region, let me call that region A. Carbon dioxide concentration is the limiting factor. But beyond that region, let me use some other color to show you this. So beyond the region A, this other part now where you're having from A all the way to the end of the graph, carbon dioxide concentration can no longer become the limiting factor, because an increase in carbon dioxide concentration does not impact on the increase in the rate of photosynthesis. So other things like your temperature or your light intensity would be the rate of will be the limiting factor in region B. But as far as you are concerned, 0.02 is still within the region of A, so that's why carbon dioxide concentration would be our limiting factor. Alright, I hope you are getting 1 or 2 knowledge about these questions. Let's move on to question number 9. The next one says a student wrote some statements about the palisade mesophyll and the spongy mesophyll tissues. The first statement says the spongy mesophyll tissue is near the stomata for gas exchange. Second one says only the palisade mesophyll cells have chloroplast for photosynthesis. The third one says the palisade cells are closely packed, but there are air spaces in the spongy mesophyll tissue. Okay, so they're asking you now which statements are correct descriptions of the 2 tissues. Which of them are the correct? So let's pick the first one and analyze. The spongy mesophyll tissue is near the stomata for gaseous exchange. That is correct. Alright, so let me try to show you this diagram of the leaf so you can just quickly use this to answer that question. This shows the internal structure of a leaf, and as you look at it very well, this is the palisade mesophyll, this is the spongy mesophyll here, and then this is the epidermis, the lower epidermis. Now the first option said that the spongy mesophyll is close to the stomata for gaseous exchange. So if you notice now, the spongy mesophyll region, the whole of this region is very close to where you have the stomata. So the first option was very correct in that regard. The second option says only the palisade mesophyll cells have chloroplast for photosynthesis. Only the palisade mesophyll cells have chloroplast for photosynthesis. That is also wrong. That's wrong because the spongy mesophyll layer contains chloroplast, but they are fewer in number, not as much as what you have in your palisade mesophyll. So that statement is wrong. The third statement says palisade cells are closely packed, but there is an air space in the spongy mesophyll tissue. So from the diagram we looked at a while ago, that is also very correct. If you look at it now, the palisade mesophyll layer is closely packed, the cells are closely packed, and this is an adaptation to have a wide surface area to be able to trap enough sunlight for photosynthesis. But you can see all these air spaces in the spongy mesophyll cells. All these are air spaces that allows for what? For gaseous exchange. So the third statement is also very, very correct. So looking at that closely, you will see that the best answer to this question is option C, that says 1 and 3 only. Alright, moving on to question number 10. What is the role of bile? Bile, A option, it contains enzymes that digest starch. That is not very correct. B option, it helps in the emulsification of fats, increasing their surface area. That is very correct. It kills bacteria in food. That's wrong. And then it improves, it provides the optimum pH for pepsin activity. That's also what? Also wrong. Correct answer to this is option number what? Option B. Now, if you want to, let me just throw a little bit light on emulsification. If this is a very large starch molecule, after emulsification by bile, it will break it down to small globules. We call it small globules. Okay, globules, globules, and this is where your lipase will now be able to do what? To act on the fatty substance. So this process I just showed now is emulsification. This is the large fat molecule, and then this is now the small globules for what? For the lipase enzyme to do what? To act on it. So take note of that. Alright, moving on to the next question. Where in the alimentary canal is the enzyme trypsin found, and what are the products of the reaction trypsin catalyzes? Looking at it very closely, you'll see that trypsin is found in the duodenum. So in the small intestine has the duodenum as the first part, it has the ileum as the last part, and it has the jejunum as the middle part. So we are correct to say what? That trypsin is found in the duodenum. But what are the products? The products in enzymes, you have the substrate, you have the enzyme, and you have the products. So the products are what the enzymes would have broken down the substrate into. So looking at it closely, trypsin breaks down protein to form amino acids. So the answer is what? The answer is A. Option number A is correct for number 11. Alright, so let's move on quickly to the next question. Number 12. What is a structural feature of xylem vessels? Xylem vessels are found, or they are responsible for the transport of water and mineral ions in plants. Basically, they are made up of lignified walls. They are very hard vessels, and then they are empty, somewhat like this. Okay, so you have it something like this. They are just continuous walls, and then they help to transport water, and then your mineral ions like your nitrates, your phosphates, etc., up the plants. Now what happens is that they do not have mitochondria, so made up of cells joined end to end. This is correct. The end to end joining forms the continuous walls, and then presence of cross walls, this is wrong, and then they have thin walls, also wrong. So the correct answer here is option B. Alright, so moving on to the next question, we are making progress. 13. During the growing season, tomato plants produce flowers and fruits. Which parts of the growing plants are sources and which parts are sinks? Now there's a concept in Igcse biology that talks about sources and sinks. So a source is where a substance is produced, and then a sink is where that same substance is taken to. So we are taking it there, or where it is used up. Okay, so having that in mind, let's see. Option A says that the flour in this case would be a sink, because when your glucose is produced from photosynthesis, where is glucose produced? It's produced in the leaf. So the flour is going to be a sink because the glucose is transported to the flour for growth and development, and also the same thing to the fruit for storage. So the best option there is option A, produced in the leaf from photosynthesis, taken to the fruit for storage, and then taken to the flour for metabolism. Alright, so moving on to the next question. Which statements are correct descriptions of methods of preventing pathogen from entering the body? Now, this is a question in the transport system, and let's see the options we have there. Fibrin is converted to fibrinogen to form a mesh. Okay, and then phagocytes produce antibodies and platelets clot blood. Lymphocytes engulf pathogens. But it asks us particularly which statements are correct description preventing pathogen from entering the body. So the best answer to this is that what? Platelets clot blood. You know, one of the functions of the platelets is that apart from clotting blood, they also prevent pathogen from entering the body. And then if we are to use this now to talk about the little process that happens during clotting, that's where we now begin to bring in the fibrin and then the fibrinogen into it. So just to throw a little light on that, let me quickly show us that. So looking at this now, this is a broken blood vessel. You notice here that the blood vessel has been broken at this point, and then that breaking of the blood vessel will now attract platelets. So platelets will begin to attract to that site. When they get to that site, a particular hormone called thrombin would help to convert your soluble fibrinogen will now be converted. Let me write it down. Your soluble fibrinogen will now be converted to this guy, which is insoluble fibrin. The insoluble fibrin is now what is going to use in making up this mesh that will block the pathogens from entering. Remember what we asked the other time, and then will also prevent the blood from and prevent your body from losing too much blood. Alright, so that was just to throw light on the procedure for blood clotting. Moving on to the next question, number 15. Which statement describe a double circulation? This is a very simple recall question. A double circulation is a process where blood will have to pass through the heart 2 times to complete a circuit. So blood passes through the heart twice to complete what? A circuit. This is as against single circulation that refers to where blood goes through the heart once to make up a circuit. So the correct option to this is option C. Question number 16. 3 statements about immunity are listed. The first one is that specific antibodies are produced in response to specific antigens. The second one is that pathogens have specific antigens. And the third one is that a vaccination stimulates an immune response. Which statements are correct? Looking at it critically, specific antibodies, yes, produced in response to specific antigen. That's very correct. They need to have the same shape. As a matter of fact, the shape of the antigen must be complementary to the shape of the antibodies. So that is correct. Number 2, pathogens have a specific antigen. That's also very correct. There are specific antigens for all the pathogens that affect the body. And then number 3, a vaccination stimulates an immune response. That's also very correct. So the correct answer there is A. 1, 2, and 3. Also, by reason of throwing more light on this, so this is like a pathogen now. This shows a pathogen, and these are antigens. Antigens are protein molecules on the surface of the pathogen, and you can notice this is now the antibody. So you see the shape of this antibody would be complementary to the shape of this antigen. It has to fit in properly so that can be destroyed by the process of immunization. Right, so let's move on to the next question. Number 17. Which muscles in the breathing system contract during inspiration? It's a very simple question on the respiratory system or gaseous exchange system. So basically, during inspiration, which is taking in of air or inhalation, the external intercostal muscles will contract and the diaphragm will also contract. So So that is the answer to that question. You can go back and check when the external intercostal muscle contracts, the diaphragm contracts, the rib cage would have to move what? When the diaphragm is contracting, let me explain that. You have two situations. This one that is curved now is a relaxed diaphragm. So in this case, the diaphragm is relaxed. But when the diaphragm is contracting, it will tend to do what? To flatten. So a flattened diaphragm, it's a contracted diaphragm. Take note of that. When this contracts, it allows for more space into the lungs so that air can flow into the lungs. Alright, next question. Which process requires energy from respiration? Take note that diffusion and osmosis are examples of passive transport. And in passive transport, they don't have to do much work. Okay, so substances don't require energy from respiration in passive transport, so those are the wrong options. The correct answer would be protein synthesis, because protein synthesis is an anabolic process, and in anabolic processes, it means a building up reaction where you have small amino acids forming your large molecule or your large polymers, which in this case is a protein. So that is a process that will require what? Energy from respiration. Moving on to the next question. Question number 19. What is produced by anaerobic respiration in human muscles? In human muscles, when you are running or you're carrying out a vigorous exercise, what happens is that the body switches temporarily from oxidation or from aerobic respiration to anaerobic respiration. In this process, the body produces a substance called lactic acid, and lactic acid is toxic to the body, so it's quickly broken down immediately after your exercise ends, and that's why you're still taking large chunks of oxygen after running or after exercising, as the case may be. And that oxygen you are taking down to break down that lactic acid is what we call the oxygen debt. So you can check it out. Oxygen debt is the extra oxygen taken in to break down the accumulated lactic acid during anaerobic respiration. So the correct answer there was option C. Alright, moving on to the next question. Question number 20. Which statements are correct for a healthy person? Blood leaving a glomerulus has the same concentration of glucose as blood entering a glomerulus. Okay, there is no glucose in the fluid leaving a nephron. Number 3, a nephron reabsorbs all of water and some of ions filtered out of the blood by a glomerulus. Okay, so let's see the statements that are correct there. The first one, let's analyze it. Blood leaving a glomerulus has the same concentration as blood entering a glomerulus. That's not true. They don't have the same concentration, because when the blood is coming to the glomerulus, it contains substances like urea, contains glucose, contains water, contains salts. Okay, all these substances are filtered into the glomerulus, into the Bowman's capsule, in a process called ultrafiltration. But then, when the blood is leaving, none of urea will be taken back, because it's a waste product, it's a chief waste product. All of glucose will be reabsorbed, some of salt and water, depending on the osmotic condition of the body. So the first statement cannot be correct. The second statement says there is no glucose in the fluid leaving the nephron. Of course, that's very correct, because the glucose is very essential to the body. So the body cannot afford to lose glucose in excretion. So if you're a normal person and you're a healthy person, there shouldn't be any trace of glucose in your urine. Number 3, a nephron reabsorbs all of water. That's why I have a problem. All of water and some of ions filtered out by the glomerulus. It doesn't reabsorb all of the water. Some of the water still comes out back as urine. Okay, so that's why option C, or the third option, is also not correct. So from there, you've seen that only 2 is correct. Moving on to the next question. When moving from an area of low light intensity, take notes, to an area of high light intensity, how do the circular and radial muscles of the iris react? Okay, so let's look at this closely and let's see something here. This is a simple question. This question is about a concept called the pupil reflex. Pupil reflex is a reflex, of course, is any involuntary activity that aims at protecting the body from getting damaged. So if you've ever entered a hall and then maybe there was no light, everywhere was dark, and all of a sudden somebody just turned on the light. You did not expect, you didn't see the person. There's that flash of light that just enters into your eye. That is what we are trying to explain the biology behind it here. Whenever that happens, in a high light intensity, let me try to do something to show you and make it a bit more better. Alright, so trying to explain that with this now, you can see that in the bright light situation, you're having a very small pupil. The pupil is this space here. The black space you're seeing in here is the diameter of the pupil. So you can notice that in bright light it is smaller as compared to what you have in dim light. In dim light the diameter is wider, and that's because in bright light, take note of this, you can write it down, the circular muscles will contract and the radial muscles will do what? Will relax. And in dim light, the circular muscles will now relax and the radial muscles will contract. As a matter of fact, these two muscles are called antagonistic muscles. Antagonistic muscles in biology, they are muscles that act in opposite direction. When one is contracting, the other is what? Is relaxing. So take note of that. I've just given you two examples of antagonistic muscles, okay, or one example rather, circular muscles of the iris and then the radial muscles of the what? Of the iris. So that is what happens. So let's go back to the question and see how we can pick up the answer. So looking at the question again, you're seeing here that when moving from an area of low light intensity to an area of high light intensity, how do the two of them react? We are moving from an area of low light to an area of high light. So the correct answer is going to be option B. And option B says that the circular muscles will do what? Contract, while the radial muscles will do what? Will relax. And then because now the size of the pupil has to be small. When it is smaller, it will prevent that flush of light from entering the eye and thereby preventing damage to the retina and the inner parts of the eye. Moving on to the next question. The graph shows the change in the percentage of infections caused by antibiotic-resistant bacteria in one country. Now, you're having on the y-axis the percentage of infections caused by antibiotic-resistant bacteria, and then on the x-axis you're having the years, and this span between 1981 and 2001. Which statement could explain the change in the percentage of infections caused by antibiotic-resistant bacteria? Looking at it clearly, the use of antibiotics has resulted in natural selection for resistant bacteria. You must know what natural selection means. The ability of this bacteria now to become immune or to develop what we call resistance. They are able to resist the effect of these antibiotics. So let's see if that is correct. The use of antibiotics has resulted in natural selection. How did you know that? If you look at the percentage of infections caused by antibiotic resistance bacteria by natural selection, now it has increased. So I think option number 1 is very correct. Okay, so it has increased over the years. 1981, it kept on increasing and increasing. So option A, the first statement, is correct. The use of antibiotics has caused a decrease in viral diseases. Now this is out of it entirely. Why? Antibiotics are not used to cure viral diseases. Okay, they would be, as a matter of fact, if this even happens, it's going to help to increase the resistance in bacteria. Okay, and then number 3, the use of antibiotics, when not essential, increase the percentage of antibiotic resistant infection. This is also correct. When you are using the drugs when you're not supposed to take the drugs, you are actually helping to develop the resistance of that bacteria. Okay, so that's why you must always be taking drugs that are prescribed by a physician. You can't just wake up and then decide that you want to take this drug, you want to take that drug by yourself. Self-medication is actually evil, and it will help to develop this resistance of superbugs in your system. So looking at that closely, the correct answer is option B. Statement 1 was correct and statement 3 was also correct. Moving on to the next one. A gardener has a strawberry plant that produces large sweet fruits. Now, how does a gardener grow most strawberry plants that all produce large sweet fruits? What they are trying to test here is your ability to understand what is called artificial selection or selective breeding. In selective breeding, you are the farmer. The farmer is inducing what he wants to see. As opposed to the first question where we saw natural selection, you don't have anything. Natural selection occurs naturally by nature. But this one, in this question number 23, is caused by you. You are the farmer. You want to see plants that will produce large and sweet strawberry fruit. So what do? The best answer there is by using asexual reproduction to produce offspring with no genetic variation. Okay, so basically what you do, you bring them together, use asexual reproduction, because the daughter or the daughter cells or the offsprings are going to be a clone of the parent cell. So the best option to our answer is A option. Okay, moving on to the next question. Right, moving on to the next question. Where does fertilization occur in a plant? The first option there is the oval, second is the pollen grain, pollen tube, and stigma. Of course, it is common knowledge that fertilization is a fusion of the what? The male gametes, of which in this case is the pollen nuclei, fusing with the what? With the oval nuclei. Okay? And where does this happen? It happens down the what? The oval. So the correct answer is option A, happens in the oval. Okay, the next one, the graph shows the concentration of two hormones during pregnancy. Now, this is estrogen, very important hormone in the female reproductive system, and the next one is hormone X. So you can see that they are both increasing. Which organ secretes the most hormone X in the late stages of pregnancy? Of course, these hormones are secreted by the ovary at the start, but as the pregnancy goes on, it now gets secreted by the placenta. And if I may add an extra information, you can type in the comment section, what do you think hormone X is going to be? Alright, if you have typed that, hormone X is going to be progesterone. Okay, progesterone. And this helps in the development of the what? Uterine lining or the endometrium during pregnancy. Right. The next question, a person has 28-day menstrual cycle. On which day is the lining of the uterus thickest? Okay, this is also similar to the first one they asked us there. So this one now, you need to understand the menstrual cycle. You need to understand the stages in the menstrual cycle before you can answer this kind of question. So let's quickly see how we can understand that. Alright, so this is a little diagram to show you the stages, the different stages in the menstrual cycle of a female. Now you're having the first hormone here, follicle stimulating hormone. You can see it's highest between 0 to day 4. This first stage is what we call the menstruation stage. Okay, so you're having it there, and then what is happening here is that the walls are being shed away. That's why you're having that blood flow and all of that. And then after that, immediately you now have the estrogen. It's getting highest, and then it's coming here to help to repair the broken walls. So it's getting that now, and this happens between day 4 to somewhere around day 13. On the 14th, that's where you have ovulation taking place, and the chiefest hormone that takes charge on day 14 is the LH, or the luteinizing hormone. So that's where ovulation takes place on the 14th. But as you go beyond the 14, from the 14 all the way to 26 thereabout, you're going to have a high concentration of what? Progesterone. And this is very easy, and this is very interesting. Why? It's getting increased because it is helping in the development of the uterine lining. You can see that the lining is getting increased. Perhaps there is fertilization, so that the embryo would be able to find a place to get implanted. So that's why it's making this place ticking. If nothing happens at the end of the day, the cycle comes back to what happens here on the 0. It begins to shed off again, and the cycle continues and continues. So having this now, day 21 would have a highest level of progesterone. So let's go back to our question and see. So the answer here is very easy. On which day the lining will be thickest? The thickest lining was, from what we saw there, was on day 21. Moving on to the next question. It says, which statement describes the sex chromosomes in a human gamete? Now, every human gamete contains 2 types of sex chromosomes. The sex chromosome is the 21st pair, and then for every female, if you are a female, every female has 2 pairs of the X chromosome, so she'll be XX, and every male has an X chromosome and a Y chromosome. So let's check out the one that's correct. X contain either an X or Y chromosome, so this statement is wrong. X cells contains only X chromosome, this statement is correct. Sperm cells contain either X or Y chromosome, this statement is correct. And then sperm cells contain only, this statement is wrong. Okay, so looking at that closely, you see that the correct answer is statement 2 and statement 3, and the correct answer there is option C. All right. Okay, so moving on to the next question. The next question says, which statements describe the effect of meiosis on chromosome number? Very interesting as well. Okay, effect of meiosis on chromosome number. So we're going to see this now. In meiosis, let me just give you a little recap of what happens in meiosis. You are going to have a parent cell that is going to produce 4 haploid cells at the end of the day. 1, 2, 3, 4. But the unique thing here is that this cell is diploid, but all the cells are what? Are haploid. And what do I mean by that? These cells contain half the number of chromosomes in the parent cell at the end of the day. So if this contains 4, each of these will have 2, 2, 2, and 2. That's how you call it diploid and what? And haploid. As opposed to what is happening in mitosis. In mitosis, if you're having a cell that had 4 chromosomes at the end of the day, at the end of mitosis, you have 2 daughter cells, and each of them will still have 4 and will have what? 4. So that's why we say that mitosis occurs in what? In diploid cells, meaning that the cells are the same. They contain the same number of chromosomes as the parent cell, but in meiosis, haploid cells are formed. So let's find the option. Looking at that very closely, you see that the correct answer is B, a diploid parent cell producing haploid daughter cells, from what we saw here in this diagram. Moving on to the next question. A female with red-green color blindness has a child with a male who does not have the red-green color blindness. The child is a male. Look at it though. They've given us everything here, so we have to be very careful. A female with a red-green color blindness has a child with a male who does not have a red-green color blindness. The child is a male. What's the chance of this child having a red-green color blindness? So we have to work this out. You need to understand this and we have to work it out. Very interesting. So let's find the female. First, you need to understand what color blindness is. Color blindness is a sex-linked trait, and sex-linked traits are traits that are always located on the X chromosome. So let me try to play this out on a different platform so we can have some way to write. Okay, so we say that color blindness is always what? Is an example of a trait located on the X chromosome, and because it's located on the X chromosome, and then you know before now that every woman has 2 copies of the X chromosome, whereas a man has just 1 copy of the X chromosome and a Y chromosome. Because even though this color blindness and all the genes located on these X chromosomes are recessive genes, and from your knowledge of genetics, you know that recessive genes are genes that are always expressed when they are together. But because of this exception that you have just 1 copy of the X chromosome in males, even though they are just a copy, an allele of it, it will be expressed in males. So due to this, they are always more common inwards in males. Let me take it a step further to explain so you understand better. For every woman, you will have 3 possibilities. So let's use color blindness for instance. You have this, you have capital X and capital X. This is capital C and capital C. This will be a normal woman. This woman is going to be a normal woman. And then for a man, the second of the female, you will be capital C, and then you have a small c. This woman is going to be a carrier woman, meaning that she has the ability to give birth to a child that is colorblind, even though she is not phenotypically expressing it. And then finally, you have this last option, small c and small c. This woman is going to be a what? A colorblind woman. She's going to be colorblind. So it means that in every 3 ratio, you have 1:3. But let's check the males. The males have just 2 options. So the male is going to have X capital C and Y, which is going to be a normal man. This man does not have it. And then you're going to have the unfortunate part again, which is going to be X small c and Y. You see, so this man is what? Is colorblind. This means that in the males you have a ratio of what? 1:2. So it means that if you have this and you have this in females, of course you should have it more common in the males than in the females. So going back to our question, I just had to do that so you can understand. So going back to our question, we have the woman to be colorblind. So this is going to be the woman, capital X small c, capital X small. Now meanwhile, I'm using this because this is how we represent our sex-linked traits. The X represents the X chromosome and the C is actually the allele. So that is crossed with a man. Let's say the man does not have, okay, the man does not have it. So the man is going to be X capital C. Oh, let me try to make this really pronounced. Let me make the capital very pronounced so that I won't be confusing you with this. So let me try to make the capital very pronounced. So I'm going to have X capital C, let me write like that and this. So if I put this in the Punnett square, I'm going to have this now as my little ratio. I'm just doing a rough sketch now so you see. So I'm going to have X capital small c here for the woman, another X small c here because she has it. And then for the man, I'm going to have X capital C here, and then I'm going to have Y. So let's do this together. This is going to be X capital C from the man and X what small c from the woman. Here also I have X capital C from the man and X small c from the woman. So here I'm going to have X small c from the woman and Y. That's the only option. Okay, the Y from the man. And here also I'm going to have X small c from the woman and Y. So remember, this question is talking about the males. They say the child is a what? They say the child is a male. They now ask you, what is the chance of this child having a red gleam color blindness? So looking at the two males now, this was a small c, don't forget. Another small c, don't forget. So it means you have 2/2, which is what? 100%. So that's why this is going to be the answer to that question. Moving on to the next question. What is a source of genetic variation that alters the base sequence of DNA? Of course, any genetic variation that would alter the base sequence would have to occur at the genetic level. So that is a gene mutation. Next question. Which technique is not used to conserve endangered animal species? Remember they say animal in this case. So we're not going to just pick anyone, but pick the one of animal. And then let's see them. Artificial insemination is for animals, and then captive breeding also is for animals, in vitro is also for animals, but they say which technique is not used for animals, so seed banks are in plants. So that's why the correct answer is option D. I hope you are following along, and I hope I am helping you to get one of those things. Alright, so let's move on to the next one. Which flowchart shows how energy is transferred from the sun to the organism? This is just very simple. This is asking us about the process of photosynthesis. And then it's a very easy one. Chloroplast helps in trap or contains chlorophyll that helps to trap sunlight. And then that sunlight is processed into glucose, and then eventually it is stored in plants. The correct answer is option D. From your chlorophyll to your glucose to your starch stored up in plants. That is the correct answer. Moving on to the next question, which statements about a food chain are correct? Now, the food chain is a simple feeding relationship that shows the transfer of energy from one organism to another. So I'll just have this to be my simple food chain. From your grass, I'll go to my grasshopper, and then I'll go to my, what should I use there? What praise on grasshopper? I'm just going to say my small bird. So I'm going to say my small bird. Alright, so let's pick this up and see. Between trophic level 1 and 2, energy is passed from a herbivore to a carnivore. Between 1 and 2. This is trophic level 1, trophic level 2, and trophic level 3. So between 1 and 2, the energy is passed from a herbivore to a carnivore. That's not correct. This grass is a producer and is not a herbivore. So that is a producer. So that option is wrong. Now, between trophic level 2 and 3, energy is passed from a primary consumer to a secondary consumer. Of course, this guy is a primary consumer, so that's correct, and this guy is a secondary consumer, so we tick 2 as being correct. Between trophic level 3 and 4, energy is passed from a secondary consumer to a tertiary consumer. So we stopped at trophic level 3. Let's add the fourth one. Let's just turn this to a big bed or a large bed. Permit me to just put it here, large bed. So this becomes the fourth trophic level. So they say between 3 and 4, energy is passed from secondary consumer to a tertiary consumer. Let's see. 3, of course, is a secondary consumer. This was producer, primary consumer, secondary consumer, and tertiary consumer now. So that is also correct. So 2 and 3 are correct. So the correct answer to question number 33 is option C. Moving on to the next question. Which process in the nitrogen cycle is involved in the breakdown of amino acid in living organisms? Okay, so normally amino acids will have to be broken down in a process called deamination. And deamination simply means that the nitrogen containing parts will be removed and transported, transported to the liver, where it is used to make urea. The urea will now be sent to the kidney, where it is excreted out of the body. So the answer is A. Question number 35. The diagram shows part of a carbon cycle, yes, which arrow represents photosynthesis. So looking at it very closely, let's look at the diagram. This shows the carbon cycle. A means that the composers are releasing carbon dioxide to the environment, so this will be decay. So A will be decay. B means that animals are sending carbon dioxide to the environment, so that will be animal respiration. Animal respiration. That's going to be B. C means that plants are also releasing. Remember that the arrowheads are the ones giving me the direction now. So C means that plants is also sending carbon dioxide to the atmosphere, and that of course is going to be plant respiration. So C is equals to plant respiration. And then D finally means that the plants are taking up the carbon dioxide. You can see that now. Plants are taking it out. And then that will represent what? Photosynthesis. So D is equals to what? Photosynthesis. Alright, so that becomes the answer. The answer becomes D. I hope you are following through. Alright, so moving on to the next question. The graph shows how the size of the population changes over time. What comes to the question? Which row identifies the phase of population growth? So looking at it very closely, you see that A is called the lag phase. We are going to explain it in the next video because we don't want to make this too long. Number 1 is the lag phase, but in the lag phase now, they're just getting acclimatized. The organisms are just getting used to the system, so that's why nothing much is happening there. In the second one, the log phase, this is the phase of exponential growth. Everything is happening, and then the organism is having a nice time. The third one is called the stationary phase. In this phase now, the death rate is equal to the birth rate, and then this is because some toxic substances are getting accumulated in the medium, and the organism is not doing very well again. And finally, the D part is the death phase. And this phase now, the phase of the rate of death is higher than that of production. So it means that the cycle is gradually coming to an end. So let's see the one that identifies the correct answer. From there, you see that D is the best. So D is death phase. We got that. B2 is the exponential phase. Either you call it the log or the exponential phase. Log or exponential, take note of that. While A is the lag, and then 1 is the lag phase. So that's the correct answer. Moving on to the next one. Eutrophication results in the reduction in the dissolved oxygen in streams and lakes. What causes the reduction of dissolved oxygen? Of course, the best answer here, looking at it, is because of the aerobic respiration of the decomposers. What happens in eutrophication is a concept where sewage is washed into water bodies. You have a farm that is close to a water body, and then you just apply fertilizer and there was rain. So rain just pushed everything straight into the water body. And then because the fertilizer contains nitrates, the nitrates will help the algae to bloom. And that's what we call algal bloom. When the algae blooms, eventually what happens is that they will block the oxygen, so the water plants will not be able to get, they will block sunlight from access to the water plants. Because they cannot get the water plants, they will die. Now microorganisms have enough food to eat and enjoy themselves, so they come, they respire, and that now causes the eventual death of the animals in the water. So that is called eutrophication. You can check it out and try to make some notes on it. And then if you have questions, you can drop them as well for us in the comment section. See how you can take care of that. Now the next question is number 38. What are the reasons for using chemical fertilizers in food production? To reduce the use of herbicides, to increase crop yields, to reduce competition between crops. It's a straightforward question, and the best answer to this is option B. You don't have a problem with that question. So let's move on to the next one, number 39. Why is pectinate used in food production? Basically, pectinase help to break down pectin, and then they break down pectin to simple sugars like glucose and the rest of them, and this is found in fruits. So that is because more juice can be extracted from the fruit. So the correct answer there is B. Remember, pectinase is an enzyme. Whenever you have ASE, it means that an enzyme is involved there. So it breaks down pectin to simple sugars. And then finally, number 40, the diagram shows the stages in the production of human insulin. Which stage uses the enzyme DNA ligase? Okay, this is very interesting as well. Now, if you've done your biology basic very well, you know that this is called a plasmid. So this is called a plasmid, and is found basically in all bacteria. They are made up of extra chromosomal materials used in genetic engineering to make insulin and penicillin, any drug or any product of it. Now, the stage that uses ligase enzyme is the option C. Why? Because this is called the recombinant DNA. Why is it recombinant? Because at this stage now, it is a combination of two different organisms, genes from two different organisms. The first one was from what? From the bacteria, which is the plasmid, the part that is appearing white. But the last part that is a little bit dark in color is the part that is the insulin gene that is gotten from a human cell. So what you use in joining these two together is the ligase enzyme. Alright, if you got value from this video, kindly help us to comment, like and share, as this will help to push this video to some other persons so that it can increase the reach. Kindly watch out, as we'll be doing more reviews on this and much more to help you do well in your exams. I pray you will do very well in your exam and you come out in flying colors as you prepare and study. Thank you very much. See you next time.